me. I have a lot of things on my mind. All right, so we'll start from here. All right, pretend you didn't hear anything I said. We'll start over. Okay, so we're talking about solute transport in cells. And again, this will be a review for many of you guys. But what we will look at is, as many of these properties that dictate the movement of solutes, how it can really make us understand the movement of metals, and specifically metal compounds, through the body and into cells. And essentially, when we're thinking about movement, we have to think about the movement through membranes. So here's our representation of a eukaryotic cell. Here we have an electron micrograph of biological membranes. All right, so we have what constitutes the plasma membrane, and we have the other components of a cell. When we're thinking about what constitutes the plasma membrane, or essentially what constitutes membranes in general, but here specifically the plasma membrane, because you have the exterior that's facing, the extracellular part, and then you have the interior that's facing the cytoplasm, and everything else is within your cell. We have this complex series of molecules, but we're all well aware of the basic component of the cell membrane, or the membranes, consisting of this lipid bilayer, where the exterior has these phosphor groups that have a particular set of properties that allow for hydrophilicity. And then you have this very greasy lipid bilayer, and the properties of that, how that can really control what type of things can actually move through. But nestled in between this lipid bilayer, we have a series of other lipids, and we also have proteins. So you have proteins that are exclusively in the exterior, so they're on the peripheral side, or you have those proteins that can actually cross through the membrane. They're known as the transmembrane proteins, all right? So what we're gonna talk about is a bit about the features of the biological membranes, and what about these features can really dictate movement. All right, so as I was mentioning, when we're looking at the biological membranes, we're essentially focusing on a complex lipid-based pliable structure that is composed of a variety of lipids and proteins. So we have a ratio of 40:1, okay? And so some of these membrane lipids and proteins are glycosylated. And if anyone has worked with glycosylation of molecules, what that essentially entails is that you're adding a component that can potentially increase the hydrophilic property of the given structure. And for proteins especially, that can allow for solubility, right? That's an important property for certain types of proteins, especially when those that may be involved in transport. All right, all cells have a cell membrane, which basically separates the cell from its surroundings. Eukaryotic cells have various internal membranes, when you want to think about them, the organelles, that divide the internal space into compartments. Mammalian red blood cells, so erythrocytes, do not have organelles. And presumably, this is because they want to make room for a lot of hemoglobin. And if you know what hemoglobin essentially is, right? Hemoglobin is an iron-based protein that is the reason why we're able to breathe. The iron in that protein will bind oxygen. Remember, oxygen is nonpolar, so it doesn't really have much solubility in our blood, which is predominantly water-based. And so the coordination of that oxygen on the iron will facilitate movement of that oxygen. But what are the basic function of these biological membranes as they pertain to the cells? So they play many important functions. They help to define external boundaries of cells, all right? So we're talking about the cell membrane there. And they regulate the movement of ions and molecules across that boundary. So we're going to talk a bit about what are those factors. They also aid in cell-to-cell communication. Again, we're focusing with the cell membrane, all right? And in signaling. All right, so typically what you have are receptors that have some ligands or substrates that will basically bind to the receptors, and that will send off a chemical signal that will aid either whether they're supposed to intake something into the cell or facilitate certain interaction with other cells. The membranes also divide the internal space into distinct compartments. That is to segregate processes and components, the organelles that I mentioned. And they serve to organize complex reaction sequences and cellular processes. So something that's extremely important is this idea about the compartmentalization, but also the general concentration, because what can happen within the cellular environment, because it's so compact with not just structures, but a number of proteins and other biomolecules, is that you can have what are known as localized concentrations, right? And that localized, really compact environment can facilitate certain interactions that may not be favorable in the absence of that compartmentalization or that increased localized interaction. So because of that, you have a number of processes that are facilitated. For instance, energy transduction, okay, biomolecular synthesis, and so forth. So oftentimes, two things that may interact within the cell environment, if you try to isolate them and study their binding interaction, you may not actually observe it, all right? And that really it speaks a lot about this idea of localized concentrations. So focusing on the common features of eukaryotic membranes, all right, so here we have the membrane consists of two leaflets of lipid-based monolayers. Okay, so one of the leaflets will face the cytoplasm. Basically, you're facing in, and then the other would be facing out, right, to the extracellular environment. So in terms of the basic structure, these are sheet-like flexible structures, 3 to 10 nanometers thick, right? So very small, obviously. The structures within the membrane bilayer are stabilized by non-covalent forces, especially hydrophobic ones. Membrane bilayers are largely composed of phospholipids. The polar heads on the exterior form a hydrophilic surface. Okay, the fatty acyl chains in the interior, they form basically a fluid environment. All right, so you have this greasy environment that allows for lateral motion, but it also can facilitate motion through the membrane. And you also have other lipids that are nestled in between. Then I alluded to this idea of these series of other structures, these proteins that are part of the composition. So if you want to break it down to two components, you have those that are the peripheral proteins and those are the integral proteins. Those that can actually span the whole bilayer. Well, what can distinguish these two? So the peripheral proteins, they are loosely associated, right? And they have these polar head groups. And what I mean by loosely associated is that in terms of if you were trying to break up the membranes, these would probably be easily dissociated from the actual membrane and probably have some capacity of being solubilized. But then when we're talking about the integral proteins, they span the lipid bilayer. You would typically see they have a large alpha helical or even beta barrel structure. Different domains positioned in different parts of the membrane based on hydrophilicity and hydrophobicity. So these tend to have a really interesting complex series of structures depending on what component of the actual transmembrane protein that you're focusing on. Now, they're not loosely associated with the membrane, okay? And in fact, oftentimes, it's very difficult to try to isolate a membrane protein because they tend to aggregate and be very difficult to solubilize. So it can be quite complicated to study. You often need detergents in order to be able to have them in soluble form. Okay, another big feature of the membranes is their asymmetry. Some lipids are found preferably within the membrane, some are found preferably external. And as a consequence of that, you will typically find the carbohydrate component of these moieties located externally. Another really big one, and that can really dictate movement of things through the membranes, is that they are electrically polarized. So what facilitates this polarization are the actual structural properties of a molecule. So whether you have charge groups, and essentially you're looking at from outside to inside, this delta of approximately negative 60 millivoltage, right? So they are electrically charged, and that can help facilitate movement. We'll see that in a moment. Now, the really important thing is that they're self-sealing, so there is no loss of membrane continuity, because if you do actually break that the seal, right, there basically would lead to lysis, and then your cell would die. And then they are selectively permeable to polar and charged solutes. We'll look at that. Specific transporters allow for this transport or to facilitate things that may not normally be able to be permeable. All right, so let's explore membrane transport, and we're going to lead this discussion as it pertains to membranes. So we'll start generally. Okay, so here is an overview of different types of transports. And so the most simplest one is the diffusion process, is movement of a molecule just simply through the membrane without needing any energy or any facilitation from an actual molecule. So a simple diffusion, we're talking about nonpolar compounds only. So they're moving down a concentration gradient, and we'll look at that in a moment. Then we have those type of diffusions that are actually facilitated. Okay, and so those would be corresponding to movement along an electrochemical gradient. Remember, I did say that the membrane is polarized. Then you have those movements that are either of the form of a primary active transport. So they're coupled to a process that's going to give them the energy, that boost, to facilitate the movement. You also have the secondary type of active transport. Here, you're moving against a certain gradient, and it's being facilitated by the thermodynamic movement or the thermodynamic drive of the movement of something that's in the direction of the electrochemical gradient. So something is facilitating this movement. Okay, so you're also getting that active transport, or that active support. Then you have these structures that are known as channels, okay, that can also move things. And here is an example of just an ion channel that allows for an ion to be able to move. We also have things that are known as ionophores, all right, that can bind. So basically, you can think of them, if you want to think about a metal, that will chelate a metal, right? And that its function solely is to bring that metal into the cellular environment. But the actual, whatever's facilitating this movement is not actually taking much of a role other than just simply the transport process. Okay, and then to the right, we have what is known as endocytosis. So all these processes that I've just described does obviously involves the membrane. But here you have a direct process in which the membrane actually forms a structure around whatever solute is coming in. So here we have just a cartoon representation of the endocytotic process. And in that process, remember, there still has to be a continuity that's maintained. All right, so let's talk about a bit about the selectivity in terms of the permeability of a cell. So all living cells interact with their surroundings by transporting solutes in and out as needed for biosynthesis, metabolism, cell proliferation, and whatnot. The transfer process is restricted by the physical and chemical properties of the solute. And so generally speaking, we're talking about these four main components: the size, the hydrophobicity, the charge, the concentration. So here I provide you a general table that gives us some indication about solute permeability, okay? And so generally speaking, nonpolar molecules will likely go through simple diffusion through the membrane, okay? But I put a little asterisk here, right? Because I will say that it's not just any molecule that can, but generally speaking, if something's gonna go through simple diffusion, we're really talking about nonpolar molecules. Then you have examples of some small uncharged polar molecules, right? You see their molecular weight, urea, water, and ethanol, that can actually go through, so they are permeable. But then when we're talking about large uncharged polar molecules and something like glucose, which we don't really think of as a big molecule, but that's big enough that it does not have the capacity to just be permeable through the actual cell. You have to have some transporter to facilitate that movement. Charged polar molecules also not permeable, and ions are not permeable. So when we're talking about this asterisk here for the nonpolar molecules, here we have size limitations. Generally speaking, the bigger something becomes, the less permeable they will be. In drug design, so I would say the concept of what will constitute something that is druggable or could be used for a therapeutic, people aim to synthesize things that are so permeable for the purpose of oral delivery that fall in the category of less than or equal to 500 grams per mole, right? This is the concept of the Lipinski's rule of 5. You will learn this in medicinal chemistry. But I would say more and more, there is evidence to show that you can actually go beyond some of the size limitations that the Lipinski's rules indicate in terms of facilitating permeability, but even in terms of facilitating things that may be orally available, right? So here I provide you a reference for you guys to look at to really get an understanding of things that have the capacity to potentially be orally bioavailable. All right, so two general routes for membrane transport. So you have the passive transport, right? And the simplest one would be the simple diffusion. So this will be in the direction of the chemical gradient. It's applicable to nonpolar molecules and again, very, very small number of small polar molecules. But then you have those that are diffusing, but they're going to be in the direction of the chemical gradient. And we're going to talk about what is the chemical versus the electrochemical gradient. Applicable to polar molecules and ions. So charge has a component here, or the polarity, and membrane protein facilitated. Now, to contrast with passive transport, we have active transport, and that is diffusion against the electrochemical gradient, right? So the key thing, whether it's in direction or against, and this will be a membrane protein facilitated. All right, so when we're focusing on passive transport, in accordance with the second law of thermodynamics, molecules tend to spontaneously assume the distribution of greatest randomness and lowest energy. So that dictates the component of the chemical gradient. So focusing on simple diffusion here, we have this cartoon visualization of a membrane, okay, that's essentially dividing two different chambers. So we have one side where we have a certain concentration 1 versus concentration 2, okay? And so basically, when we're talking about simple diffusion here, we're looking at a net movement of electrically neutral solutes across a plasma membrane toward the side of lower solute concentration, okay? So there is this drive that is being directed by a difference in concentration. And so you reach a point where you will no longer have movement because you reached an equilibrium, okay, where you have basically reached an equal amount of concentration of that given solute. So now your concentration 1 is equal to your concentration 2. So that's not to say necessarily that the movement has completely stopped, but basically that you have reached a point where you're going to have this balance in charge, right? And that is a concept of moving in the direction of the chemical gradient from higher concentration to lower concentration until you reach an equal amount of the given solute. Good. So something that is related to that, right, in terms of this idea of the chemical gradient, but you have another component, which has to do with the charge. So this is the electrochemical gradient. And so essentially you have a net movement of electrically charged solutes, which is being dictated by a combination of the electrical potential of the membrane and the chemical concentration difference across the membrane. So in our cartoon on the left, what we have is the two chambers separated by the membrane. We have the two species there. One is negatively charged, one is positively charged. So on the left side, what we have is here a higher amount of the negatively charged species. On the right, we have a higher amount of the positively charged species. So in terms of concentration, you have then this movement of the negatively charged species from the left to the right, because they're going from higher concentration to lower concentration. Likewise, for the positively charged species, you will have this movement, a chemical gradient movement, from the right to the left, from high concentration to the low concentration. But coupled with this as well is this idea of a charge balance, because you're going to reach a point at equilibrium where not only have you balanced the amount of these species from one side to the other, but you also have balance of charge. So you have those two components, the charge balance in addition to the concentration. Okay, so the electrochemical potential reaches 0 at equilibrium. So in passive transport, the transported species always moves down its electrochemical gradient and is not accumulated above the equilibrium concentration. Right, so this is something that is, in terms of the Gibbs free energy, so delta G, will be a favorable process. In active transport, the transported species always moves against its electrochemical gradient and is accumulated above the equilibrium concentration. Okay, so that's the big difference here. So active transport is, technically speaking, thermodynamically unfavorable and takes place only when coupled directly or indirectly to an exergonic process. So in other words, that unfavorable process has to be coupled with something that is favorable in order to be able to be facilitated. So an example of what can facilitate this is the breakdown of ATP, because of breakdown or the phosphate lysis of this molecule can lead to a liberation of energy that can supply the energy needed to move that other species against the electrochemical gradient. You can also have a concomitant flow of some other chemical species down this electrochemical gradient. So, in other words, that species is moving along the gradient, so that's thermodynamically favorable, and is allowing this thermodynamically unfavorable thing to move against. All right. So then, as part of this, right, this idea of something facilitating, right, you have then transport of a polar or charged solute. And so here's where you can start thinking about metal complexes, so charged metals, charged metal complexes. So there are membrane proteins that facilitate the diffusion of polar charged solutes called transporters. And these transporters, they are also known as permeases. And so if you guys remember biochemistry, anything that has the suffix ase implies that it is an enzyme. But these are special types of enzymes. And what I mean by special type is that you're not actually changing the structure, or you're not transforming a molecule into something else. You're basically facilitating the increase in the rate of movement, right? So these enzymes are not like enzymes in the traditional sense. So they can facilitate in the direction of or against the electrochemical gradient. All right, so they provide a polar charge environment to facilitate a lower energy transition in intermediate state. All right, so let's look at that. What does that actually mean? So let's look over here as a representation. So let's imagine we have a charged species, or let's say something that is polar, charged or polar, whatever, that's interacting with the with water molecules, right? So you have either these polar contacts, or you have a ion interaction, all right, with the water molecules. Well, you have this equated environment surrounding that specific species. But for that species to be able to move through, right, let's say passive diffusion, what has to happen is you have to have a loss of that interaction between the water molecules and that species. You're going from basically what would be a polar environment to something that is not, right? Okay, so what happens then is you have energetic penalties. There will be this activation barrier for the loss of the water molecules, but in addition to that movement of that species in that environment, then actually may not be favorable. To then, when it gets through to the other side, to then be facilitate this interaction with these water molecules. So the reaction coordinate that we see here, okay, that's essentially what we're looking at here, this reaction coordinate profile. We're looking at the free energy, and we're looking at the starting point of the energy, and then these activation barriers that are actually pretty high, okay? And then that's associated with the loss of the favorable interaction with the water molecules in that species, and then the movement through the actual membrane, okay? And then finally, when you get through the membrane, and then you again start to have these water molecules associated with that species. So in terms of energy, from before and after, there technically is no change in energy. But there is an activation barrier, and that activation barrier is what affects the rate of the movement, okay? So then you have potential transporters that can help to, minimize the amount of energy penalty. So a transporter that, because of the residues within the transporter, may facilitate a charged or polar environment that's favorable for the movement of that solute. And as a consequence of that, you then bring down the relative energy of your transition state, or even your intermediate, okay? And as a consequence of bringing down the energy, or the activation energy, that's going to facilitate the kinetics. Your kinetics is going to be much faster, this movement here. All right, so when we're looking at this, the delta G of the transport, okay, that is going to be much less than the delta G of the simple diffusion. Okay, so this will be applicable when you think about metals as charged complexes or as charged species. All right, now, since we're now alluding to this idea of transporters, okay, so they're classified in terms of the movement of the solids and also the directionality. So you have the uniports, as the name implies, uni, one solid moving through, and then you have those that are co-transporters, the symport, that are moving two things in the same direction, or the antiports, moving two things, one in the opposite direction of the other. Okay. Now, excuse me. The directionality can be with or against the electrochemical gradient, and so transporters can facilitate both passive and active transport. So when we're talking about primarily active transport, the energy released by ATP hydrolysis drives solute movement against an electrochemical gradient. So again, as I said previously, ATP, it consists of these 3 phosphate groups, all right? And so the loss of the phosphate group, let me just give us a little visual here. The cleavage 1 phosphate group for giving us ATP and then generating ADP, and then inorganic phosphate, okay? Well, that liberates a lot of energy. And that is an energy amount of negative 30.5 kilojoules per mole. In fact, when you study biochemistry, you know that ATP is basically a storage of energy for us. All right, so then you have this lysate, and you have that energy, and that supplies the ability of the solute to move against its electrochemical gradient. So you're moving, low concentration to higher concentration. Here we have a representation of the secondary active transport, a gradient of ion, S1, that has been established by primary active transport. Okay, so you have primary active transport. So solute 1, right here, this track solute 1, is moving here and is going against its electrochemical gradient. And what's facilitating that is ATP lysate. Okay, so the phosphate lysis. Okay, so you have that. So that now solute 1 is moving. Then you have now provides the energy to drive a co-transport of a second solute, S2, against its electrochemical gradient. All right, so what do I mean there? Well, let's keep tracking. Solute 1 now is going in this transporter and is going along its electrochemical gradient. It's going from high concentration to low concentration. And now it's enabling solute 2 to go against its electrochemical gradient from low concentration to high concentration. All right, so that's how we can see where you have a series of these interacting network of transport. So this general classification of transporters, right? So then the transporters divided into the category of those that are carriers and channels. And then in the category of carriers, we have then those that are the uniporters, the secondary active transporters, or primary active transporters. All right, so look at this. All right, so here we're looking at transporter kinetics. And so what I am alluding to here is something that you discussed in biochemistry, this idea of the dependence of a velocity on a concentration of a substrate. But here, right, if we're thinking about a transporter as an enzyme, increasing the rate of movement, normally when you think about enzyme kinetics, you're thinking about an enzyme speeding up the rate of the conversion of a substrate to a product. The same idea. And in fact, the data that you obtain can be fit using the Michaelis-Menten equation. Okay, so I'm providing you that information. So you have the same type of profile in terms of the movement of something. So here what we're looking at is the movement of zinc, the zinc uptake. And the zinc, what's happening here, is being moved by a transporter in this yeast. And then we're seeing how the rate of the uptake is dependent on the concentration of the zinc here. And so the profile that we get is how much the transporter is increasing that rate. So what we're looking at is the, so here is being done with a vector. So you have a vector control that does not have the transporter. And so you see that in terms of the zinc uptake dependence on the concentration, there's really not much of an increase, right? So you see the saturation curve, but you don't have a really dramatic increase in the rate. However, when that transporter is overexpressed, meaning that you have a lot of the concentration of that transporter, okay, what you're going to see then is the saturation curve, okay? And a saturation curve can imply that you're basically saturating the amount of sites available for the given, in this case, a given metal ion to be moved. So the saturation implies that you have maximized the amount that you can actually reach. So the rate equations for transporter kinetics can be derived exactly as for enzyme catalyzed reactions. So you typically will use what is known as initial rate kinetics, yielding an equation comparable to the Michaelis-Menten equation, resulting in this hyperbolic plot or this saturation curve, right? And one of the key features of this equation is this value here, the T, okay? That's not a binding constant. Basically, it gives you a parameter of the relative enhancement of the rate of movement, right? And that occurs at a concentration of the substrates, in this case, the zinc, when you have half the maximal velocity. And the maximal velocity is this parameter here at the saturation of that curve. Okay. All right, so let's focus on channels. Remember, that's part of the categorization of the transporters. So proteins that form pores in the membrane for diffusion. Pores open in response to a signal. Okay, so what are these possible signals? You can have a membrane electrical potential change, or you may have ligand binding. Okay, what are these processes that are facilitating that? So here I'm showing you this cartoon representation about whether there is an electrical signal or whether there is a substrate that binds to the transporter, to the channel, right? And that binding interaction is going to imply that the channel needs to open up in order to facilitate a movement. One of the features of these channels is they do not have high stereospecificity, and they catalyze transfers at rates several orders of magnitude greater than carriers. They actually approach the diffusion limits. So we're talking about pretty high rates, so pretty high K values in terms of the movement of species. And they're not saturable, all right? So what does that mean, not saturable? So basically that plot that I was showing you before, you wouldn't get necessarily the saturation curve. And that's probably because there is probably necessarily no physical constraint in terms of movement of the solids through. All right, but here we have an example of carriers, so proteins with high substrate stereospecificity. So they bind substrates at one side of a membrane. Okay, so here's a representation of a glucose transporter. And there are many glucose transporters. This is just a representation of a binding process in a pocket. That binding interaction of the substrate will then send the signal that now the actual carrier has to open up and then facilitate that movement through. So there's a recognition process of the substrate. Okay, so it catalyzes transport at rates that are below the diffusion limits. And you will have saturable-like enzymes. Why saturable? Because you're going to have so many sites available for binding of the glucose to facilitate there. So you can maximize the availability of the interaction. Okay. Then you have those things that are known as ionophores. These are a diverse class of organic molecules that increase the permeability of membranes to particular ions. They can be peptide or non-peptide based. Bacteria are known to produce peptidic antibiotics that inhibit the growth of other organisms by shuttling and depleting the levels of important ions. And we're actually going to talk a little about this. This goes into the concept of what is known as nutritional immunity, which basically has to do with trying to protect, so say an organism protecting itself by holding on to certain essential elements and basically keeping them away from predators. Here we have a visual of an antibiotic that forms a complex with potassium. Okay, so this is a monocation here. So you have the linomycin will interact with the potassium, the cell wall. It'll pick up the potassium and it'll facilitate the movement into the inside of the actual bacteria. Then we have what is known as receptor-mediated endocytosis. So this is what I was saying to you. All of these processes involve the membrane, but here we actually have the physical features of the membrane becoming part of the actual motion, of the actual movement. So in general, endocytosis is the process by which cells absorb molecules by engulfing them using their plasma membrane. This process is often coupled to a ligand binding to a receptor, which triggers delivery of this ligand into the cell. All right, so we just have this visual over here. And so you have these green protein receptors, okay, that will recognize the substrates here, these star substrates, That recognition results in a signal that now the membrane has to undergo this endocytotic process that will basically lead to the formation of what is known as an endosome. So this is basically the feature of an endosome. Sorry. This process can be clathrin dependent. Clathrin is a large protein that forms a coated pit on the inner surface of the plasma membrane. The pit fuses to the membrane and forms a coated vesicle in the cytoplasm. So the importance of clathrin is that it serves as a protecting agent. It basically marks the endosome and says that, look, whatever's inside here, okay, the receptor I'm talking about, the protein, we actually want to recycle. So we only want to move that solute into the cellular environment, but after this process, we want to bring that receptor back, okay, bring it back so that it can be recycled to undergo more processes of bringing other amounts of that particular solute. Okay, so the clathrin serves as a protecting group. Then you have those clathrin independent processes where you don't have this protection and the endosome is proteolyzed and everything inside of it will get basically dispersed into the cell. Okay, so that just gives us an overview of this idea of recycling receptors in certain processes, and in other processes that's not necessarily a requirement. All right, so thinking about this collectively, let's talk about this idea of mobilization. Mobilization of metals for transport. Okay, so what does mobilization mean? Well, basically that has to do with converting metals into a bioavailable form that it can be useful. Okay, and so now you obtain it into a form that now you can use, let's say, a biomolecular vehicle to then allow for it to be transported. So there are three key ways that organisms mobilize metals to make them bioavailable. One of them is chelation. Essentially, you transform metal into a stable but moderately labile formulation. All right, what does that mean? Stable, we all know what that means. You form a stable compound, but moderately labile. Well, moderately labile refers to the ability of that metal to then easily exchange. And that's something that you want, because you want to be able to capture the metal, but you don't want to be able to capture it and then you can't use the metal. You want to be able to liberate it. So you want it to be relatively labile, so it can exchange where needed. Another process is redox, where you reduce or oxidize metals to an appropriate oxidation state for specific transporters. And the third is acidification. You solubilize metals at relatively high levels using acid, so you prevent formation of hydrolysis products that are typically poorly soluble. All right, and I emphasize this. Hydrolysis products is one of the worst things when it comes to working with metals. So if anyone does metal synthesis or try to make a metal compound, many metals, if you work with them in a neutral pH or a basic pH environment, they can be quite difficult to work with. They may even precipitate. But oftentimes, metals are quite favorable to work with under acidification conditions or acid conditions because they can remain soluble. So that is a process that organisms take advantage of. We're going to focus on this idea of chelation, but we're actually going to see these three processes as we make discussions of mobilization of metals in general. Chelation. All right, so here we're looking at certain biomolecules that are well-known chelators known as siderophores. Okay, and so these are used by bacteria, fungi, and some plants and marine organisms for acquisition of iron. There are hundreds of known siderophores, and many new ones are still being discovered. And they typically feature the iron-binding moieties catechol. And so catechol is a group like this. Okay, we have these two OH groups. All right, so this is your basic component of a catechol. Or the hydrazamic acid or hydrazamate, depends on whether it's positively charged, whether it's negatively charged or neutral. So this will be in the anionic form. And so it's a structure just like this. Okay, so both of these have the capacity to serve as bidentate, excuse me, let me draw this better. Okay. Let's go back into here. Didn't mean to do that. Let's go. And here we go. All right, so both of them have the capacity to metal bidentate. Okay, so here the metal would be iron. I mean, that's primarily what these siderophores are useful for. They do have, obviously, the capacity to bind other metals. They're not necessarily strictly iron binding, but they're utilized for that purpose, to mobilize iron. So here we have the representation of the enterobactin siderophore, and this is the desferrioxamine B siderophore. And if you're aware about iron chelation therapy, okay, these, especially this one right here, are quite used for the treatment of iron overload. All right, so here I give you a visual of the iron siderophore structures. All right, so I'm going to talk a little bit about what we see here. So typically, monomeric structures of 1,1 metal ligand hydrogen form. All right, so that's a typical way of categorizing metal ligand interactions in terms of the number of metals, the number of ligands, and the number of hydrogens. So here, when I say 1, 1, and in this case 0, that means that the ligand has no protons bound to it, protons at a site that coordinates the metal. And so what you have is a stoichiometry of 1 metal to 1 ligand. Okay, so if we're looking at the iron siderophore structure, well, how does that fit? Well, we have the coordination of iron, and that's represented here as the green symbol here. That's your iron. There's only one iron, and it's coordinated in a manner that involves 3 of the catechol moieties in the enterobactin, so that you have a coordination number of 6. Okay, coordination number 6 because you have 3 catechol metal interactions. Okay, but still, that's still just one ligand interacting with one metal, and the interaction sites, the oxygens, well, those protons that were originally there will no longer be there because of the iron coordination. And we see something similar for the ferrioxamine, where you have a coordination number 6 compound because you have 3 of those hydroxymates as bidentate ligands coordinating your metal center. And then the oxygens from those actual moieties are not there. They have been deprotonated. One of the ways that people typically characterize the relative affinity of siderophore for metals is using this parameter. So here I show you a table where we're looking at binding constants. Okay, so we will actually look at this in a little bit later in a different lecture, this idea of what does beta mean. Okay, beta is basically the equilibrium constant, so a K constant, but the beta is used because typically when metal and ligands interact, it can involve more than one type of interaction. I'll talk about that in a moment, okay, or I'll talk about it in a later discussion. So when we talk about beta, just think about it as a binding constant. The higher that binding constant is, the stronger the interaction is. So the enterobactin has an extremely high affinity constant for iron. So you have a log beta of 49. But there's another parameter here, parameter PM. And when you think about PM, it's kind of like thinking about pH, right? When you think about pH, that tells you about the relative concentration of protons. Well, here is very similar. You think about relative concentration of metal, but here it's metal that's free. Okay, so PM equals to the negative log of the concentration of the metal in solution in water is a measure of free or uncomplex metal left in solution. Okay, and that gives you a gauge of how much, another way of measuring the affinity. So the higher the PM, the lower the amount of iron that's going to be in solution. All right, so that's a way that you can actually compare one type of siderophore versus another. All right, so the outer membrane receptor proteins for ferrosiderophore. So what recognizes these molecules? Because these molecules are produced, right? Bacteria will produce them, and they will send them out into the surroundings to start capturing iron, whether it's capturing iron and stealing it, maybe even from us, right? We have a number of bacteria that invade us, right? So there are receptors that can recognize these complexes. So bacteria have evolved high affinity outer membrane receptor proteins for binding the iron siderophore. These are substrate-specific carriers that engage in energy-dependent active transport of the iron siderophores across the outer membrane. And so here we're going to talk about the gram-negative. Okay, so that it has a particular composition in terms of its structure. All right, so here we're talking about a gram-negative bacteria. And so the outer membrane receptor, or one type that can recognize as iron siderophore, is the following, where we have this FHUA, ferrichrome iron complex. So you have this transporter, or this receptor that will recognize the iron complex, iron siderophore complex, coming from E. coli. Okay, so the structure of that protein consists of 22 strands, is a membrane spanning, and is anti-parallel beta barrel. So that's the structures that you see here. Okay, and so what's been captured here is the actual iron complex bound to the transporter. So the apo form of the protein has a conformation that blocks or corks the periplasmic side. But then when it comes in contact with the iron siderophore, that triggers a conformational change that now opens up the transporter to facilitate movement through. Okay, so once released to the periplasmic space, the complexes, we're talking about the iron siderophore complexes, are bound by a high affinity periplasmic protein. Transport of iron across the cytoplasmic membrane can be in the ionic ferric or ferrous form. So ferric implies iron 3+, ferrous implies iron 2, so either form, or as an ferric siderophore complex using what is known as an ATP binding cassette transporter system. All right, so what does that mean? So you can have movement of the iron. Once it gets facilitated, that movement gets facilitated by that transporter as iron, as an ion, whether iron 3 or iron 2, so no ligand surrounding it, or as the iron siderophore complex. So that complex keep maintaining the iron to facilitate this movement. Very interestingly, ferric siderophore receptors in organisms that do not produce siderophores exist. So there are organisms that actually have evolved these receptors that can recognize iron siderophores. So basically, they are scavengers. So they don't produce their own siderophores. Rather, they take advantage of the molecular machinery of other organisms to then capture iron. So they're mooching off of another organism. They're taking the shortcut. They're not doing the work of making these siderophores. They're like, let's just evolve these receptors, and we'll recognize and take the iron that we need. So some organisms feed off of others because of a lack of an optimal method of chelating iron. So here is an example of a yeast that preys on bacteria and have evolved transport systems for different ferric siderophores. So that's something that can exist, where one organism exists, amongst other types of organisms, and can recognize their molecular machinery and be able to adapt, to evolve, to be able to exploit the machinery of another organism. And so that it has been observed, and that allows for movement of iron so that they can exploit the metal themselves without having to do all the steps that we've talked about. All right, everyone. So that wraps up our introduction to this idea of transport. In our next lecture, we're going to talk about transport of iron as it pertains to the human body. All right, everyone, have a great weekend, and I'll see you next week.
Thank you. Have
Professor.
a nice weekend.
Yeah, go ahead.
I didn't see last class's lecture in the website. Is it uploaded already, or?
Well, yeah, it's been
me
uploaded
and
since.
missed it?
Yeah, I think you just missed it. It's been uploaded for a while.
Okay. Thank you.